Today we are going to learn about early development in C. elegans and vulva development in C. elegans. So when we try to learn about the structure of C. elegans or the differentiation in C. elegans, we could see portions like pharynx, ovary, intestine, vulva, rectum, anus, etc. And the actual size is less than 1 mm. Then when we try to elaborate the structures, we could see oviduct will be there, spermatheca will be there, and then sperm storage region, uterus will be there, vulva is there, spermatheca will be there. Later meiotic zone will be there, ovary is there, mitotic zone will be there, distal tip cell will be there, sperm will be there, oocytes are there, embryos are there. Okay, so nematode roundworms like a good group for studying embryology because they have a relatively small number of chromosomes and a small number of cells with invariant cell lineages. C. elegans are 1 mm long. Free-living soil nematodes have a rapid period of embryogenesis, about 16 hours in petri dish. Adult form is hermaphrodite, producing both egg and sperm sac containing exactly 959 somatic cells, and the entire cell lineage is traced through its transparent cuticle. The C. elegans genome was the first complete sequence ever obtained for a multicellular organism. The zygote of Xenorhabditis elegans exhibits rotational holoblastic cleavage. During early cleavage, each asymmetrical division produces one founder cell, A, E, B, M, S, C, and D, that produce differentiated descendants, and one stem cell, P1 to P4. In the first cell division, AB is the anterior founder cell and the posterior stem cell P1. So hypodermis, neurons, pharynx, other, EMS, MS, muscle, pharynx, neurons, somatic gonad, others. Then intestine, muscle, hypodermis, neurons, germline, muscle will be there. Okay. Abbreviated cell lineage start. Anterior posterior axis formation. When the sperm pronucleus enter the oocyte cytoplasm, the centriole associated with the sperm pronucleus initiates cytoplasmic movements that push the sperm pronucleus into nearest end of the oblongous oocyte. That end become the posterior pole. Then the sperm provides a protein CYK4 that activates GTPase in the egg cytoplasm and alter or activate egg actin microfilaments to reposition PAR proteins, pushing them anteriorly. The most important entities localized by PAR proteins are the P granules, appear to be a collection of transcription regulators. The protein of these granules include RNA helicase, poly A polymerase, and translation initiation factors. After fertilization, the random scattered P granules move towards the posterior end of the zygote, so they enter only the P1 blastomere formed from the posterior cytoplasm. Through division, ultimately the P4 cells containing the P granule, whose progeny become the sperm and eggs of the adult. The localization of P granules require microfilaments, not microtubule. And the cortical cytoplasm contains PAR3 and internal cytoplasm contains MEX5. Cytoplasm flow towards sperm pronucleus that contains CYK4. PER2 replace PER3 protein near sperm astus. The domain of PER2 expression expands as the sperm nucleus moves towards the center. Then protein polarity same. After first division, AB blastomas has MEX5 and PAR3, while most of the P1 blastomeres has very little MEX5 and has a cortex of PAR2. Then formation of the dorsal ventral axis. The dorsal ventral axis of the nematodes is seen in the division of the AB cell. As the AB cell divide, it becomes longer than the eggshell as wide. This squeezing also causes the ABP cell to take a position above the EMS cell that result from the division of the P1 blastomeres. The ABP cell defines the future dorsal side of the embryo, while the EMS cell marks the future ventral surface of the embryo. So controls of blastomere identity. C. elegans demonstrate both the conditional and autonomous modes of cell specification. The P1 cells develops autonomously without the presence of AB, generate the posterior half of the embryo. The AB cell isolation makes only a small fraction of cell types that it would normally make. Therefore, the specification of the AB blastomere is conditional, need to interact with the descendants of the P1 cells in order to develop normally. Then autonomous specification. The determination of P1 lineage appears to be autonomous, with the cell fate is determined by the cytoplasmic factor like P granule that specify germ cells. Also, the SKN1, PL1 and PIE1 protein encode TFS that act intrinsically to determine the fats of cell derived from the 4 P1 derived somatic founder cells MS, E, C and D. EMS cell divide, but only one progeny MS have maternally expressed SKN1 protein that determined the MS cell develop autonomously to pharyngeal tissues. Embryos with homozygous SKN1 mutant have extra hypodermal fate tissue in place of mesoderm and endoderm in the embryo. SKN1 is a maternal protein and it activates the transcription of MED1 and MED2. The MED transcription factors specify the entire fate of the SMS. MED genes in other cells can cause non-EMS cell to become EMS even if SKN1 is absent. PL1 activity is needed for normal development of the somatic descendants of the P2 blastomeres. Embryos lacking PL1 have no somatic cell types like C and D. MEX3 protein inhibit the translation of PAL1 mRNA and SKN1 also inhibit PAL1. PIE1 placed into P blastomere through the activation of PAR1 protein and it appears inhibit both SKN1 and PL1 fraction in the P2 and subsequent germ cell line fate. Mutation of the maternal PI1 gene result in germline blastomere adapting somatic fates. P2 cell behaving like EMS blastomere. PIE1 preserves the totipotency of the germ cell lineage. Internal zygote E fate mesoderm pharynx. Conditional specification. Conditional specification can be seen in the development of the endoderm cell lineage. The EMS cell requires a signal from its neighbor of the P2 blastomere. If P2 cells are removed at early 4-cell stage, the EMS cell will divide into 2 MS cells and no ectoderm will be produced. During specification of MS cells from EMS cells during maternal SKN1 protein, the POP1 signal block the pathway to the FE fate by blocking the ability of SKN1 activated MD1 and MD2 to activate the TBX35 gene. TBX proteins activate the mesodermal gene in the pharynx, PHA4 and muscle HLH1 of C. elegans. The P2 cell produces a C. elegans homologue of WNT proteins, the MOM2 peptide which is received by the MOM5 protein in the EMS cells. The results of the signaling cascade is to downregulate the expression of the POP1 gene in the EMS daughter destined to become the E cell. The POP1 deficient embryo, both the EMS daughter cell become E cells. The P2 cells is also critical in giving signal that distinguish the ABP from its sister ABA. ABA give rise to neurons, hypodermis and anterior pharynx cells, while ABP makes only neurons and hypodermal cells. If reverses the positions of two cells, their fates are similarly reversed and a normal embryo is formed. In other words, ABA and ABP are equivalent cells whose fates are determined by the position in the embryo. The interaction between P2 and ABP is important for specification of ABP fate. If P2 cell is killed at the first celled stage embryo, ABP cell does not generate its normal complement of cell. The ABA cell can also be made into an ABP type cell fates if it is forced into contact with P2 cell. The interaction is mediated by the GLP1 protein on the ABP cell that serves as notch receptor and the ABX1 protein on the P2 blastomere serves as delta ligand. GLP-1 mutant have ABP is transformed into an ABA cell. This ABX1 signal break the symmetry between ABA and ABP because it stimulated the GLP-1 protein on ABP plastomium. The next major topic, vulva induction in C. elegans. In the early development, they are male and gonad produces sperm which is stored in for later use. As they grow older, they develop ovaries. The eggs rolls through the region of sperm storage, are fertilized inside the nematodes and then pass out of the body through the vulva. The organ forms during the larval stage from the 6 cells called the vulval precursor cells, VPCs. The cell connecting the overlying gonad to the vulval precursor cell is called the anchor cells. The anchor cell separates the Lin3 protein, a paracrine factor that activates the RTK pathway. If the Lin3 gene is mutated, the VPC will not form a vulva, instead become a part of the hypodermis skin. The 6 VPCs influenced by the anchor cell from an equivalent group. Each member of the group is competed to become induced by the anchor cell and assume 3 fates depending on its proximity to the anchor cell. The cell directly beneath the anchor cell divides to form the central vulval cells. The 2 cells flanking that center cell divide to become the lateral vulval cells. 3 cells away from the anchor cell generate hypodermal cells. If the anchor cell is destroyed, all 6 cells of the equivalent group divide once and contribute to the hypodermal tissue. If the 3 central VPCs are destroyed, the 3 outer cells which normally form hypodermis generate vulval cells instead. The Lin1 protein is received by the LET23RTK on VPCs and the target cell of the kinase cascade is the Lin31 protein. When this protein is phosphorylated in the nucleus, it loses its inhibitory protein and able to function as a transcription factor promoting vulval cell fates. Mechanism of induction to develop vulva. The Lin3 protein forms a concentration gradient. The VPCs close to the anchor cells, the P6P, receives the highest concentration of Lin3 protein and generates the central vulval cells. The 2 VPCs adjacent to it receives a low amount of Lin3 and become the lateral vulval cells. The VPCs further away from the anchor cell do not receive enough Lin3 to have an effect, so they become hypodermis. The central vulval cells also signals lateral to the 2 adjacent cells through Lin12 Notch protein and on their cell membrane. The Notch signal activates the microRNA MIR61, which repress the gene that would specify central vulval fate as well as promote these genes that are involved in forming lateral vulval cells. Vulval precursors, gonad anchor cell, basement membrane, gonad anchor cell, P8P, P7P, P6P, P5P, P4P and P3P. The formation of anchor cell through cell to cell interactions. The formation of the anchor cell is mediated by the Lin12 gene, Notch receptor. In C. elegans hermaphrodites, 2 adjacent cells Z1 PPP and Z4 PPP are equivalent or both have the potential to become the anchor cell. The 2 equivalent cells producing fluctuating amount of signal, that is lag2 delta, and a receptor, that is Lin12 Notch. Reception of signal turns down lag2 production and upregulates Lin12 production. The chance event causes one cell to produce more lag2 than the other cell at a particular time. This stimulates more Lin12 production in the neighboring cells. The cells producing more Lin12 produces less lag2, so one cell is delivering the lag2 signal and the other cell is receiving it. So the cell secreting more lag2 or the signaling cell become the anchor cell and the receiving cell become the ventral uterine precursor. The loss of function Lin12 mutants, both cell becomes anchor cells. In gain of function mutants, both cells become uterine precursors.